Hi, and welcome to the metabolomics part of our session. I'm Ulle de Bruin, a scientist at DNA Genotech. I will take you through today slides on the OMNIget collection device for gut metabolomics. So why do we need a collection device for gut metabolomics? So the first point here is perhaps pretty obvious, is that if we don't stabilize a sample, the metabolomics profiles of a stool sample will undergo rapid changes at room temperature. Next point is perhaps not as obvious, is that the gold standard of collection of gut samples is freezing at -80 is not well suited for at-home collection. So it relies on cold chain, which are costly and unreliable due to temperature fluctuations associated with shipping. And there's also that safety risk if you would choose to ship on dry ice for the donor. So if you use the gold standard of collection for metabolomics, you will only reach a limited pool of donors that can be reliably recruited. So then another consideration, you might think, I can use a nucleic acid stabilization solution to perform my gut metabolomics analysis. Well, these are not chemically compatible across all metabolomics assays. And because they're usually typically also complex in their makeup, they will introduce a bias into your, particularly your untargeted metabolomics analysis. So that's why now DNA Genotech offers a novel device, OMNImet.GUT, to enable donor-convenient, large-scale fecal metabolomics with high-quality samples required for discoveries in this exciting field. And that's pictured to the right, the OMNImet.GUT device. It features a volumetric cap so that you can control the amount of stool that enters the sample. And the solution is a stabilization solution that's specifically formulated for metabolites, and then we have a mixing ball in the tube as well to help with homogenization. So this next slide shows results from our evaluation and comparison of OMNImet.GUT device compared to the gold standard. So again, -80 freezing of stool samples, and this is LCMS untargeted metabolomics results. From Metabolon Inc., so trying to detect as many metabolites as possible in a sample and profile that sample. So to the left here on your screen, we collected stool samples from 7 donors, either into the OMNImet.GUT device, that's in red, or as well as into the flash freezing of raw samples. At -80, that's the blue. And then we determine the number of metabolites that could be detected for these 7 donors. And then we see that the red and the blue, the OMNImet and the flash freezing, the number of metabolites could be detected for both collection methods were very, very similar for all 7 donors, indicating that the OMNImet.GUT device performs similarly to flash freezing in the number of metabolites that can be detected. So we don't introduce any bias into the sample, as well as we actually maintain that richness of the metabolites in the sample. And then next to your right is a correlation analysis. Here we compared fresh frozen, again, same samples, and then we held them in OMNImet.GUT for between the different colors indicate the time that the samples were held in OMNImet.GUT at room temperature, so between 0, 1, 4, and 7 days. And then these bars indicate the Spearman coefficient of all those metabolites. Here on the left is entire view of overall the 800 to 900 metabolites and the Spearman correlation. So between T0 and fresh frozen, it was the median was 0.9. So between 1 and 0 is the Spearman coefficients that they're given, and above 0.8 is very high. So we get very high correlation between OMNImet.GUT samples and the flash frozen samples. And then if you go from left to right, the different colors indicate the time points, the amount of time at room temperature that the device were held at. And then for 1 to 4 to 7 days, we don't see any change in the Spearman coefficient, indicating that the stability of the profiles overall is very high in the sample. And then we can split that up, the total metabolites into different pathways, super pathways that are indicated here at the bottom. And then for gut microbiome samples and looking at metabolites, the ones that are going to be most interest to researchers that are microbially derived are the amino acid metabolites, the lipid metabolites, and your xenobiotics. So if you look at those, they perform very similar to the overall picture as well. For example, lipids, we get between flash frozen and T0, close to 0.9. And then if you go 1, 4, and 7 days, it doesn't notably change, indicating that the metabolites are stabilized within the days for 7 days at room temperature. So, okay, that's good. So we looked at the overall metabolomic profile. We looked at the super pathway. What about if you look at the within the super pathway at individual metabolites? We can do that for lipids, for example. We can look at the short-chain fatty acids, and that's shown in this slide. We perform targeted LC-MS analysis, again with metabolome. So we looked at 8 short-chain fatty acids. Here we showed the results of 6. They all perform the same, all 8. One example is butyrate. We collected into OMNImet.GUT, held the samples for up to 13 days at room temperature and determined the concentration at 0, 1, 4, 7, and 13 days. And we can see then that the concentration of butyrate remained unchanged for the entire time of the experiment. So at least 13 days of root stability for the short-chain fatty acids. In contrast, unstabilized samples, what goes on here is that the concentration increases, indicating ongoing microbial activity in those samples. We're able to quench that microbial activity using the OMNImet.GUT sterilization solution. And then in this next slide is results from a recent publication by Ramamurthy et al. In BMC Microbiology 2021, in which researchers from Innova Children's Hospital and Metabolome performed a hospital-based field test where stool samples from 16 infants were both flash frozen and stored in OMNImet.GUT for 3 to 4 days at ambient temperature. And then samples was also collected between 2 different time points with a mean time of 22 days in between them, so that the development of the infant gut could be followed at the metabolomics level. And then we see then in the PCA to the left here of the thousands or so metabolites in the samples that were detected. So in blue is the frozen samples and red is OMNImet.GUT samples that and the different symbols indicate the 2 different time points, that the samples from the same individuals cluster closely regardless of collection method. So for example, subject 1, the blue and the red here together, and then number 5 as well, and 12, and so forth. And then the partial separation was also observed between the 2 different time points, so time point 1 and time point 2. This is consistent with both methods being able to uncover those metabolomic changes that occurs across the infant during the gut development. So I think this shows a good example of utility of the OMNImet.GUT. And again, the OMNImet.GUT performed favorably compared to the flash freezing of a sample, which is the current current gold standard. So overall, the OMNImet.GUT enables convenient at-home collection of fecal samples for metabolomics, including short-chain fatty acid analysis. We have stability of the gut metabolomic profiles for 7 days at room temperature without cold chain. And we saw here in this presentation that it's compatible with LC-MS. We also know it's compatible with GC-MS, and it's suitable for collection of fecal samples from all donors or independent of the age, including infants. So I thank you very much for your attention.